Started. So thank you all for being here late in your Friday afternoon.
Connection.
Are we connected? All right, good. I can't see anybody this time, but so as I thank everybody, I also want to extend a particular thank you to those listening in on Skype. My understanding is that Angela's mother, Judy, is online, as well as her brother, Jonathan. And they hail from the great country of Canada. So Angela is from Vancouver, and I can't really banter with her very well about sports because I'm just hopeless at hockey. She comes to us with a bachelor's degree from University of Utah, yes?
Hawaii, sorry.
BYU?
BYU, Idaho.
Oh. Should have checked my notes right before the exam, right? It's been quite a week. And I think that what I do remember about your degree is that it is essentially an individualized major, which I think is just so perfect for Angela, because to know Angela is to know a person who is passionate about so many things. And so that is one of the things that is quite special about her. Some of you may know that she's an accomplished vocalist, and that she also enjoys very much the outdoors of Hawaii in terms of water sports and hiking. And she also, little known fact, she has this amazing rare skill that she can take a somewhat dull knife and make a perfectly straight cut on a round surface, namely a lace on albatross egg. She can do this like no one else I know. So, and I think Stacy can attest to that. So, and skill sets indeed. So, as I was also thinking about remarks about Angela, she also is very good at sort of rolling with the punches and going with the flow, because as I recall, as we started off your career here at HPU, we actually had a completely different project in mind that had something to do with organic contaminants. And as our NIST friends came into the fray, and we ended up structuring and taking advantage of an amazing opportunity to work with our NIST colleagues, she was very, very flexible and amenable to taking the opportunity to really do what you're going to hear about today, which is an amazing project analyzing trace elements in marine mammals. So I am not a trace elements chemist, and so what I will do is I will place the lay onto Angela, but I want to very, very much acknowledge that her co-advisor, trace elements chemist from NIST, so Dr. Colleen Bryan. So you. And Colleen, will you want to come up and say a few words?
Yeah, I'll say so I met Angela on our first trip out here to Hawaii when we were staking things out for NIST, and we conducted a National Marine Mammal Training workshop. And she, as Brenda pointed out, with the knife for bird eggs, also excelled at using that dull butter knife on cutting blubber. So she has been very instrumental in helping out with doing the banking aspect for preparing tissues for the specimen bank when the stranding happens. And I got to know her a little bit better when she came out to Charleston and she did her lab work for a summer with us. I never realized being a Canadian would be such a foreign national issue. And I had to jump through hoops and government paperwork just to get a Canadian to be able to work at our lab. So, yeah, so Angela was very enthusiastic in the lab and just always been very welcoming when I've come out to visit. We've gone snorkeling together and had some good sailing cruise one time. And she showed me the island, so it's been nice. And when she was in Charleston, we showed her South Carolina and lots of the southern stuff. So
like alligators.
Like alligators, yes.
Thank you so much. So just a couple of final notes for housekeeping. Much like, I feel like I'm repeating myself a little bit because we were just here on Wednesday saying the same thing. But I do have to tell you that, and remind you that, in fact, this is an exam. And so this is a two-part exam where what Angela will do is deliver her presentation of about 45 minutes to deliver the main results of her work while she was with us working on her master's degree. And then there will be a time for open questions. So all of you will be invited to ask her questions about her research. And then there will be a closed-door session that will be held over in the room across the way. And so at that time, we invite you to relax and make your way over to the Kona Brewing Company in Hawaii Kai. And eventually, and it won't be 3 days, but eventually, we will meet over there to either congratulate or drown sorrows. But either way, there will be Kona Brewing Company
7 o'clock.
7 o'clock.
7 o'clock
when we're going to
on the
meet.
front patio.
So without further ado, I do want to introduce Angela, who will present her thesis entitled Hepatic Levels of Trace Elements in Stranded Pacific Cetaceans and Examination of a Biomarker of Trace Element Exposure.
All right. Thank you all very much for coming today. I realize that many of you don't know a lot of about trace elements or don't know a lot about marine mammals like cetaceans. So don't worry, by the end, you'll have a good handle on all of it. Thank you so much for coming, and do ask me hard questions at the end. I'd like to start with a brief overview of what I'll be talking about today that is in the same organization as my written thesis. First, we will do some background and talk about anthropogenic contaminants, or contaminants from humans, in the marine environment, specifically trace elements, which are the elements that I studied in my project. We will then talk about how the Hawaiian archipelago is an ideal location for monitoring these trace elements, how the marine mammals that I'll be looking at, specifically cetaceans, are excellent sentinels of trace element contaminant loads, and how trace elements of interest can be widely variable, and the ones that I'll be looking at specifically. We'll then go over Chapter 2 and look at the analytical methods that I used to measure trace elements in stranded whale liver tissues, look at some correlations and differences among life history groups that I found, then do a brief comparison to other regions, including other oceans, to see how levels in Hawaii compare to them, and finally look at some really high-level animals in our sample set and discuss the potential for health effects. Finally, for Chapter 3, I did an investigation of metallothymine, a potential biomarker of trace element exposure, and I looked at whether or not it would be a good biomarker. And so we'll be discussing my preliminary results for Chapter 3, and finally the conclusions of my research and future directions that I'll be going in and that I recommend for other researchers in this field. So, let's start with the big picture, contaminants in the ocean. Human impacts on the ocean is of increasing concern worldwide. One of the ways that humans impact the ocean is by the inputs of contaminants. Some of the contaminants that we have is nutrient loading, persistent organic pollutants, which are man-made chemicals such as pesticides and industrial chemicals, and the artificial increase of trace elements in the environment. Specifically, we are interested in trace elements in this study. Trace elements are elements that you've probably seen before. They are some of the 92 naturally occurring constituents of the periodic table. They have very unique characteristics that make them all different from one another. And what defines them as trace elements is that they have a low relative abundance in either the natural environment, such as seawater or a sediment layer, or they are required at very low levels for health and development in organisms. Because of these two distinct differences, it separates trace elements into two categories: essential and non-essential trace elements. The trace elements are all around us in the environment. Here are some of the trace elements and places that you will either be exposed to or come in contact with them. For this study, we will be looking at a number of essential trace elements and non-essential trace elements. Chromium, manganese, copper, zinc, and selenium are all essential for normal biological functions. Non-essential elements include arsenic, strontium, tin, cadmium, mercury, and lead, and potentially have the ability to be toxic. The Hawaiian archipelago is the area that I decided to do my research in. It's interesting for doing trace element and contaminant research for a number of reasons, including it's the most remote island chain in the world. In other words, it's the farthest away from continents than any other island chain. An important location already for global monitoring of climate and ocean changes. However, very little trace element literature exists for the marine environment in the Hawaiian region, and trace element research in top-level predators has not yet been done. For other reasons, Hawaii is also interesting. For example, Hawaii has some very unique geological features. It's made up of a chain of islands which are volcanic mountains. Volcanic activity is ongoing in the Hawaiian region, and these volcanic islands have steep slopes that create ideal habitat for a wide variety of resident and migratory animals. Many people assume that Hawaii is relatively untouched and unpolluted. I assumed this initially when I started researching because of its clear waters, clear air, and beautiful scenery. However, the Hawaiian Island region has experienced considerable anthropogenic impacts from contaminants from things such as pesticide use, dumping of munitions and equipment into the ocean, sewage and landfill spills that we see correlated with flooding events, the shipping industry, and terrestrial runoff from urban and agricultural areas. The next question was, what will I study these trace elements in? And the Marine Mammal Stranding Program at Hawaii Pacific University gave us an excellent opportunity to study trace elements in whales and dolphins. Whales and dolphins can collectively be called cetaceans. They share many characteristics in common with humans. They are large mammals, have long lifespans, undergo long reproductive cycles. They also have usually only one offspring at a time. They utilize similar marine resources to humans, and they are excellent indicators for contaminants in the ocean due to the fact that they have a worldwide occurrence in all oceans. They are top-level apex predators, and they are sensitive to anthropogenic contaminants and activities. Excuse me for a moment, this is getting in my way. Cetaceans can be split into two main categories: baleen whales, or mysticetes, and toothed whales, or odontocetes. As you can see, they have quite different mouths. This is a baleen of a humpback whale calf, and as you can see, this false killer whale has very large teeth. Because they have particularly different diet preferences, baleen whales tend to accumulate much lower levels of anthropogenic contaminants than toothed whales. Whales and dolphins, or cetaceans, are exposed to anthropogenic contaminants by two mechanisms, either bioaccumulation or biomagnification. Bioaccumulation is simply the accumulation that occurs over an animal's lifespan that results in longer-lived animals having higher levels of contaminants and shorter-lived animals having lower levels of contaminants. Biomagnification occurs when a substance that is highly persistent and difficult to metabolize or offload accumulates in a food web so that the substance is transferred from prey to predator up the food web, increasing in concentration by orders of magnitude with each trophic level. This process of biomagnification is particularly important for organic contaminants, such as man-made chemicals, as we heard in Mel's talk on Wednesday. However, most trace elements do not undergo biomagnification, except in the case of mercury. Methylmercury has been known to consistently biomagnify in food webs to high levels, so that top-level predators such as cetaceans can accumulate high concentrations. Mercury is a non-essential trace element, and I'd like to introduce you more specifically to the other trace elements that are non-essential that I studied. In order of atomic weight, we have arsenic, strontium, cadmium, tin, mercury, and lead. They are all collectively known as well as heavy metals. They have a number of natural sources, such as volcanic activity and weathering of substrates. Anthropogenic sources include mining, smelting, waste incineration and processing, and terrestrial runoff. The health effects for each of these trace elements is different for each one. However, they all have the ability to alter immune function, which can lead to animals that have high levels of contaminants to be more susceptible to disease and parasitic infection and tumor development. Along with these non-essential trace elements, I analyzed 5 different essential trace elements, all important for health and biological function. In order of atomic weight again, we have chromium, manganese, copper, zinc, and selenium. The natural sources for essential trace elements are similar to non-essential trace elements, mainly weathering processes and volcanic activity. Anthropogenic sources are also similar, such as mining, smelting, terrestrial runoff, etc. The health effects of essential trace elements are not common, negative ones at least. And this is primarily due to the fact that essential trace elements are very carefully regulated in tissue and easily offloaded. However, health effects have been known to occur in the case of both a deficiency and an overdose. Marine mammals such as cetaceans have the ability to tolerate natural fluctuations in their environment and in their food web, in their prey species, due to interactions between trace elements. For example, selenium has been known to mitigate the toxic effects of some of the heavy metals, particularly mercury. Mercury, quite a toxic trace element, will accumulate and biomagnify in food webs, and cetaceans primarily are exposed to mercury from their food sources. Mercury and selenium have a very interesting relationship. The detoxification pathway for mercury in liver is highly dependent on selenium. Methylmercury in a food source will be absorbed into a cetacean's body and be demethylated. In the liver, selenoproteins will bind with these mercury ions, and eventually the selenium in those selenoproteins and the mercury will bind directly to create mercury selenide crystals that have a very low solubility constant, which means they are very stable, relatively inert. These mercury selenide granules are visible here in the liver tissue of this rhesus dolphin. The little dark black spots are those granules. This interaction between mercury and selenium produces a 1-to-1 molar ratio in the livers of most adult cetaceans. It's literally a sequestration pathway where the mercury is taken out of the system and hidden away and then accumulates there in the liver. This pathway allows cetaceans to basically even high levels of mercury have been seen in animals without them showing levels of like toxic effects, thanks to selenium. My samples came thanks to the Hawaii Pacific University Stranding Program from the Hawaiian Island region and the Mariana Islands, specifically 39 from the main Hawaiian Islands and 2 from Guam and Saipan. The stranding locations specifically are shown here, the red dots indicating individual stranding locations and the large red dots indicating locations of multiple strandings. All strandings were single animal events between 1997 and 2013. 34% of my samples did come from the island of Oahu. One of the reasons this is the case is that strandings happen everywhere. They can happen in very remote locations where field sampling has to be done on site. They can also happen in areas where we're able to transport the animal to Hawaii Pacific University for a more thorough necropsy and sampling protocol. The samples for my set come from a wide variety of age groups. For example, in my sample set, we have small dolphin calves, large adult whales, and very large calf humpback whales. The size range for bodies in cetaceans is the largest range of any mammalian taxon. In the center is a little swimmer for comparison. He's not a marine mammal. And my sample set consists of 1 baleen whale, 3 sperm whales, 1 species of baleen whale, 3 species of sperm whale, 3 species of beaked whale, and 9 species of dolphins. These groupings are genetically distinct, as you can see from this phylogenetic tree. Baleen whales, sperm whales, beaked whales, and dolphins all branch separately, and there is no overlap between groups. Some groups are more closely related than others, but because there is no overlap, I was able to use these groupings in my analyses. So the objectives of Chapter 2 was to first analyze the trace element levels in these hepatic tissues, covering 16 different species of stranded Pacific cetaceans from the Hawaiian Islands, Guam, and Saipan, and establish some initial trace element concentrations. Secondly, my next objective was to identify correlations between trace elements and differences among life history groups such as age group and species group. Next, I decided to compare concentrations found in my study with those in other regions. And finally, I wanted to determine if levels of non-essential trace elements indicated a possible toxicity risk to Hawaiian cetaceans. The methods that I used, I did at the National Institute of Marine Technology Lab in Charleston, South Carolina. And I used 2 different methods. For sample processing, the first 2 steps were the same. I subsampled the liver that had been taken at necropsies and frozen. It was then homogenized to create a frozen powder that could later be analyzed. For trace element analysis of all of the trace elements except for mercury, the tissue matrix had to be broken down with an acid-assisted pressurized microwave digestion, where you add nitric acid and hydrogen peroxide and mix it with the tissue in a pressurized vessel and microwave it. The solutions were then analyzed with high-resolution inductively coupled plasma mass spectroscopy, which I will talk about a little bit in more detail in the next slide. For mercury, the frozen liver powder was weighed into weigh boats and directly analyzed on a direct mercury analyzer that utilizes atomic absorption spectrophotometry to measure the mercury. However, some of our animals did have levels above the detection limit of this analyzer, and they had to be had to use this, we had to dilute them basically. So in order to dilute them, later on they had to be acid assist pressurized, microwave digested, just as we did for the other analysis, and then were reanalyzed on the same direct mercury analyzer. And this was done thanks to Colleen, because at the end of my stay, we still had animals that were off the chart in concentration. So a number of our animals were reanalyzed later. So here's our ICP-MS and the direct mercury analyzer. Inductively coupled plasma mass spectrometry was used for most of the trace elements. The first step was to do an initial analysis using quadrupole ICP-MS, which would then allow us to choose internal standards and prepare a matrix-matched single-point standard addition method. You see, this first quadrupole ICP-MS method was only about 80% accurate, straight analyzing the samples. Once we had initial 80% levels, we were able to do this matrix-matched single-point standard addition method, where you take your sample and split it, and one half of the sample is spiked so that the trace element levels that you're interested in measuring are 4 to 5 times higher in concentration than the unspiked sample. These 2 points then allow us to measure the difference, and it's far more accurate than just straight measuring the unknown sample. It also allowed us to determine which elements would be good to use as internal standards. We specifically used yttrium, europium, scandium, and ruthenium because they're very rare and the levels were extremely low in our samples. So we also put those into our samples as internal standards at known concentrations. Finally, we were able to do a high-resolution ICP-MS run to quantify our samples by running the spiked and unspiked samples along with standard reference materials and blanks. Now, you may be wondering why we didn't just analyze mercury by the same method. Unfortunately, mercury tends to be very sticky and can gum up the works inside the instrument, and requires a lot more cleaning and preparation and time between samples. And since we had a far more simple method, the direct mercury analyzer, we analyzed mercury separately. The data that we got for our trace elements from the ICP-MS didn't come out as concentrations. They actually required quite a bit of calculation. Here is the equation used to determine the concentration from the raw data count rates of each trace element isotope in the samples. The results from the samples, blanks, and SRMs were used, and with this equation, every single trace element concentration was determined separately. This equation utilizes the count rate data of the split and spiked samples, internal standard ratios for the unspiked and spiked samples, and standard reference material numbers. For the mercury analysis, the instrumentation was a little bit more simple. The steps that the sample goes through once you load it into the analyzer is first the sample is thermally decomposed. So it's basically put into a little oven, dried, and then burned up in oxygen gas. This catalytic conversion moves the sample in the gas, and amalgamation occurs on a gold trap. Finally, once pure, the mercury is again atomized, and the concentration can be determined with atomic absorption spectrophotometry. In order to do accurate measurements, we created calibration curves with standard reference materials for low concentrations and for high concentrations. And you can see here the 2 cells of the analyzer. We analyzed the samples as tissues, or in the case that they had to be diluted, as solutions, SRMs, standard reference materials, and blanks together. The data that we got came in the form of peak areas. We used these peak area calculations from the direct mercury analyzer, along with the sample mass that we loaded, and the calibration curve equations that you can see here for low levels and here for high levels, along with the results from the blanks we ran to determine our concentrations. Finally, I was able to look at what my concentrations were like. My first hypothesis would be that essential trace element concentrations would be less variable than non-essential trace element concentrations. And that is what I found. As you can see here, with concentration on a log scale on the y-axis, and trace elements on the x-axis in order of atomic weight, the essential trace elements have blue box plots, and the non-essential trace elements have red box plots. The blue essential trace elements show less overall variability between samples than the non-essential trace elements, which show far more spread. I then decided to look at correlations and differences between life history traits. I hypothesized that trace elements would co-vary according to known biochemical interactions, as we talked about earlier, like with mercury and selenium, with shared biological mechanisms and with age-related accumulation patterns. I also hypothesized that we would find differences among age groups with high levels of essential trace elements in immature animals and high levels of non-essential trace elements in more mature animals. Finally, I hypothesized that trace element correlations would reflect known diet preferences of phylogenetic groups. So here's my correlation analysis. Now, looks kind of complicated, but let me explain it for you. The blue highlighted area is the essential trace elements, and where they correlate is also blue. Yellow is the non-essential trace elements, and where they have correlations is in yellow. And where they overlap with one another, non-essential with essential, it's green. The first correlation I found was between mercury and selenium. It is also the strongest. And it's most likely due to that biochemical interaction that we saw with mercury and selenium sequestering together. I also found a positive correlation between copper and zinc, which could indicate a shared biological mechanism, since both copper and zinc are regulated in the body by metallothionein proteins. They do, however, accumulate with age in similar patterns, as do other trace elements. Now, I was pretty impressed by how strong of a correlation we had here at 0.92 and a p-value lower than 0.001. And I decided to look a little bit more closely, and I was surprised by the result. They have a very strong correlation. Ta-da! Now you might be wondering, how is this possible? Look at this. So we have selenium concentration and mercury concentration, and the correlation between them is near perfect, with an R value of 0.9998 and a p-value that's extremely low. This is with Pearson's correlation. Now, with ecological data, we don't usually see this type of perfect correlation. And I assure you, this is real, and most likely due to the interaction we see between mercury and selenium in the liver. Now, you might be wondering who this individual is way up at the top. And it happens to be a false killer whale female, who we'll be talking about in more detail later on. Now, this interaction we see between mercury and selenium, so perfectly together, is supportive of the idea that mercury and selenium, when they sequester together, will reduce the likelihood of toxic effects of mercury in that animal. I decided to then look at age group patterns, and I started by looking at my trace element results summary. And I saw an interesting pattern in the lowest and highest individual animals. For example, we see here blue essential trace elements and red non-essential trace elements. The lowest levels of essential trace elements for 3 out of 5 elements were in adult age group individuals, whereas the highest levels of these essential elements were found in individuals from the calf age group. I found the opposite different trend in the non-essential trace elements, with individuals from the calf group having the lowest levels of non-essential trace elements, and individuals from the adult group having the highest concentrations of these non-essential trace elements. So I decided to look at these differences to see if there really was a real difference with ANOVA and other pairwise comparisons. And I found a few interactions, only 2 in the essential trace element category. If you see here, with concentration on the left, on the y-axis, and adult age groups on the x-axis, I found significantly higher levels of zinc and copper in the calf age group than in the adult age group. And as you can see here, especially with zinc, a possible trend of decreasing concentration over lifespan. I also found some differences in non-essential trace element levels. For cadmium and tin, calf groups, age groups, had significantly lower levels than adult age groups. And again, we have concentration and age groups. Concentration on the y-axis and age group on the x. Now, with mercury and selenium, I expected to see a similar interaction, since they tend to sequester together. And that is what I found, with calf and subadult groups being significantly lower in both selenium and mercury concentration than the adult age group. Just to check, I also calculated mercury-selenium molar ratios to see if there were differences between the age groups. And we found a nice, as you can see here, a nice trend increasing with age group, with calf and subadult individual groups being lower than the adult group. Now, feeling really optimistic, because I found all these great results with age group, I decided to look at phylogenetic groups. I wanted to know if there would be differences between baleen whales, sperm whales, beaked whales, and dolphins. However, this kind of comparison in such a varied sample set, 41 animals of 16 different species of various ages, it can be a complicated thing to try to do. I ran an initial comparison and just looked at the levels, and I saw that baleen whales seemed to have much lower levels of mercury than dolphins. However, as we saw from the age differences previously, we found a significant interaction between age group and species group, which did not allow us to actually do this comparison, because the baleen whales in my sample set are all calves, while the dolphins are of various age groups. So I decided to look within age groups to see if there would be differences. I looked within subadults and adults only. And I did find some more differences. Chromium levels were significantly higher, and strontium levels were significantly lower in sperm whales than in the dolphin group. And selenium and mercury were both significantly lower in beaked whales than in dolphins. And this could support the hypothesis that there is a diet-based difference between these groups. Sperm whales and beaked whales have a largely cephalopod or squid-based diet, while dolphins have a more varied diet, including cephalopods, fish species, and even other marine mammals. I then decided, since I have such a varied sample set, it would be really neat to compare the concentrations that we are finding here in this region of the ocean to other oceans. My hypothesis would be that across all of my cetacean samples, there would be some sort of signature pattern or difference that I would not find in other regions. This analysis ended up being extremely difficult due to the varied nature of my sample set. So I decided to just look in one species of dolphin, the bottlenose dolphin, which is widely studied across all oceans. And here is a comparison between maximum values found in other oceans to the maximum value in our study for cadmium, lead, and mercury. As you see here, with concentration on the y-axis and different ocean regions on the x-axis, with the far right being our sample max value for bottlenose dolphins. Cadmium levels were similar, I would say, to max levels found in other oceans. The same was found for mercury in the comparison with studies in the Western Pacific. However, lead and mercury were found to be quite a bit lower when compared to other regions in the case of lead, or regions outside of the Pacific in the case of mercury. Now, before you get too excited about there being low levels of heavy metals in the Hawaiian region, let me just remind you, this is just in a bottlenose dolphin. We had a lot more samples, and I decided to go ahead and test my hypothesis to see if there was a toxicity risk for Hawaiian cetaceans in their heavy metal levels. My hypothesis was that the non-essential trace elements in stranded Hawaiian cetaceans would be below the levels known to be toxic. However, I found quite a different result. Here we see, with concentration on a log scale on the y-axis, and non-essential trace elements, as well as selenium, on the x-axis in order of atomic weight. I've marked for cadmium the low effect level determined for southern Australian bottlenose dolphins for renal damage in liver tissue cadmium levels, and the high effect threshold for renal damage from the same animals from Lavery et al. And 13 of our animals were above that low effect level, and 2 of our animals were above the high effect level. The second highest, as you see here, is a subadult Cuvier's beaked whale, and the highest is our adult female false killer whale. For mercury, I have marked here a low effect level determined, a minimum effect level determined for Atlantic bottlenose dolphins and pinnipeds in the Atlantic. And this is the range for detoxification limit for terrestrial mammal livers. Now, 12 of our animals of 9 different species were above that 60 microgram per gram minimum level for toxicity due to mercury. 9 of them were within the range of the limit for detoxification in terrestrial mammal livers, and 1 was high above. And you can probably guess that this animal we were particularly worried about. That's a pretty high level of mercury. This animal also had the highest level of lead, cadmium, and tin, and the winner of highest heavy metals is the false killer whale adult female in our sample set. Sad winner. Let me tell you a little bit more about the false killer whales. It's actually a very interesting case study. False killer whales, this individual specifically was an older adult female. So she's had a long life to accumulate contaminants in her body. She comes from the insular population of false killer whales in the Hawaiian Island region, which has recently been designated as endangered. This population has been known to accumulate high levels of organic contaminants, and this individual specifically also had high levels of organic contaminants such as PCBs and other man-made contaminants, as we saw on Wednesday from Mel's talk. This The species also interacts with the longline fishery here in Hawaii and eats squid and large predatory ocean fish, such as mahi-mahi and tuna, just like we do. So should you be worried? Well, the worst case scenario of these high heavy metal levels is that they pose a health risk to still living populations of cetaceans, such as the other endangered false killer whales that are out there. It could also indicate that there's some sort of anthropogenic input affecting the local ecosystem, and could be an indicator of a possible health risk to local human populations who utilize the same resources or who eat whales, as some people do. However, there is an alternative. These high levels of heavy metals could be due to the fact that they are false killer whales. So I decided to do a little comparison and see if their eating habits and lifestyle lend them towards these high levels. And I found similar levels of mercury on the west coast of North America, in British Columbia, in a false killer whale stranded on Denman Island, a false killer whale stranded on Vancouver Island, and a killer whale, or orca, also stranded on Vancouver Island. Now, I was not able to determine if they had high cadmium and lead and tin levels as well, but these really high concentrations of mercury could indicate that they acquire these levels due to their trophic level and the diet that they have. So, just a quick review of my hypotheses. Essential trace elements were less variable than non-essential trace elements. Elements were found to co-vary according to known biochemical and biological mechanisms and age-related accumulation patterns. Differences were found among age groups, and higher essential trace element levels were found in immature animals and lower levels in adults, whereas high non-essential trace elements were found in mature animals. Trace element concentrations reflecting known dietary preferences of phylogenetic groups was supported by my results. However, trace element levels did not show some sort of signature pattern to compare to other regions, and non-essential trace element levels were not below the levels known to cause toxic effects. So, to investigate a little bit more into the biological effects of these trace elements, I decided to look at a possible biomarker of trace element exposure called metallothionein. It's known to be a biomarker in other animals, mostly smaller ones such as bivalves, and it is known to be responsible for the homeostasis of copper and zinc and to sequester cadmium in kidney tissues, mitigating its toxic effect. It has been found in kidney and liver samples of cetaceans in the past, and I wanted to determine if it would be a good biomarker in my animals. So the objective was to measure levels of metallothionein in my kidney and liver samples for each of my sample animals, determine if metallothionein was a viable biomarker, and do this all utilizing immunoblotting techniques. Now, I had some mixed results. But my original hypotheses were that non-essential trace element concentrations would correlate with the protein biomarker, so that animals with high levels of heavy metals would have elevated levels of metallothionein. I also hypothesized that there would be some age-related differences with metallothionein, and that they would mirror the trends seen in trace elements. Finally, I thought that we would probably see metallothionein levels higher in kidney than in liver tissues. My method was Western blotting. I had to extract the cytosolic fraction of my samples. Sorry, this clicker is just getting me. I then quantified the total protein in my samples and separated the proteins in the samples by size using gel electrophoresis. As you see here, here's the gel. I then used the Western blotting technique to transfer the proteins to a membrane, which could then be used to visualize the proteins on. And I did this through a series of steps of antibody incubations, and I used chemiluminescence to visualize the bands at the end. Unfortunately, we now refer to my results as the good, the bad, and the ugly. So this is a Western blot membrane in its final stage. Along the top we have the lane numbers, and in lane number 1 we have a protein ladder or size marker so that we can determine what the sizes of the other bands on the membrane are. In lanes 2 through 8 I loaded samples from my sample set, as well as the standard reference material QCO3LH3 from NIST, which is a pygmy sperm whale liver. In lanes 9 and 10, I loaded 2 different concentrations of a positive control, and then these were my results. As you can see, there's quite a bit of darkness in all of the bands. Ideally, we would like to see a distinct band and everything else being to be white. So this is quite a bit of background staining. The good news, in the good, the bad, and the ugly, is that we seem to have metallothionein in at least 3 of our samples, a striped dolphin and 2 spinner dolphins. This is the size that we expected to see our metallothionein on. However, our protein, our positive control is up here because metallothionein needs to be fused to a stable protein, in this case GST, in order to work in the gel. And that makes it a larger size. However, seeing these dark bands demonstrates that my antibodies worked. Now, as you can see by these dark bands at regular intervals up the lane, metallothionein also polymerizes. So it's quite possible that these bands up here, most likely, are also metallothionein. And if this is the case, then we also found metallothionein in our humpback whale, in a pygmy and dwarf sperm whales, and, well, the QCO3 did have metallothionein at both of these levels. So, now a little bit more into the bad and the ugly. Here's another example of one of my blots. This one is the false killer whale. I also found metallothionein in her kidney tissues. Now, metallothionein is a little bit difficult to work with as a biomarker. It's not your average biomarker for sure. It has a very small size. It has a relatively low abundance in tissue and is very prone to polymerization. Since it has these characteristics, it can make it very hard to detect this protein in your samples and difficult to quantify. Also, the positive control that I had is tagged, making it a different size, which makes quantification a little bit more complicated and difficult to confirm that the bands I'm seeing actually are metallothionein. And the highly sensitive detection method that I needed to use, namely chemiluminescence, produced high background and gave me some technical issues that also made my analysis more complicated. So my conclusion was that metallothionein, at this point, may not be a viable biomarker in stranded cetaceans, partially due to the variable tissue degradation levels of my sample set and the highly variable nature of my samples. Definitely further study is needed and will be done. So my first 2 hypotheses I was not able to address with my results. However, my last hypothesis that higher levels of metallothionein would be found in kidney samples was confirmed because when I ran liver samples for the same animals, I didn't get any results. So, I feel that my last hypothesis so far has been supported. Finally, some conclusions. You've made it this far, I'm almost done. Trace elements were measured successfully in 16 species of cetaceans stranded in the Hawaiian and Mariana Islands. Significant age-related differences were found, as well as species group differences. High levels of cadmium and mercury were found in some cetacean individuals, indicating the potential for heavy metal toxicity. And metallothionein, although present in cetacean kidney tissues, may not be a good biomarker candidate. Now, my future steps are to continue doing my trace element biomarker research in preparation for publication, and I'd really like to make something work. So I'll be in the lab come next week doing more biomarker work. I also have some recommendations for future research projects. I think it would be very interesting to look at stable isotope levels and trace element levels in prey items of these cetaceans to confirm the possibility of transfer from diet to these animals. Also, a histological examination of the tissues of those really high-level animals to see if there is evidence of toxicity in the tissues. Finally, it would be really interesting to compare live cetaceans to stranded cetaceans with trace element levels, and this can be done by sampling skin, doing skin biopsies on live animals and comparing it to skin in stranded animals, as we have seen in the literature in the past. Also, mercury speciation in tissues would be interesting in these animals to see what their methyl mercury comparatively to their inorganic mercury is like. Finally, I'd like to acknowledge everyone who's helped me get this far. I absolutely could not have done it without my amazing committee members. Brenda has been a wonderful mentor, helping me and being patient with me along every step of the way, and allowing me to be as nitpicky and perfectionist as I naturally am, while gently encouraging me to stay on track. I'd like to thank Christy for all the opportunities she's given me to get elbow deep in whales and learn about them hands-on. Colleen has been integral in this project. I would not have been able to do my analyses without her. She made these really difficult and technical analytical methods make sense, which is quite an accomplishment. Her encouragement and professionalism has also been an inspiration. And Andrew Green, who has always encouraged me and given me wonderful advice on just how to survive being a graduate student and to just keep going. I'd also like to thank NIST, the HPU Graduate Trustee Scholarship, and CTAC and the Marine Mammal Conference for funding support. My samples I could not have done my analysis without my samples and without the help in processing and sending them off that came from the HPU Marine Mammal Stranding Response Program and all its volunteers. And finally, who kept me sane were my lab mates and all of my friends and family, who are too many to mention, except for my loving husband. These are all the references for my presentation, and I am now ready to take questions. Yes, Jen.
Angela, part of the diversity of samples that you have in this sample set is that they were collected over about 15 years. Is there anything in the literature on these trace elements that shows temporal trends? And should you also be concerned about the difference in time collection? Could they be increasing or decreasing through the time frame that the samples were collected?
I have seen in the literature, especially in the case of mercury, decreasing trends with time in areas where less pollution is going into the local ecosystem than before. However, it would be very difficult to see such a trend in these cetaceans since the region is so remote and we don't really know where these animals are feeding and they come from such different species. If we could do a longitudinal study with just one or a few species that are very similar to one another and have similar diets, it would be interesting to see if there's a trend. However, because of decomposition, even though we've collected a lot of samples over the last 15 years, I was only able to use the freshest samples, the code 1 and 2 animals, the ones that were alive when they stranded or really freshly dead, and that significantly narrowed the numbers of my samples down. I think the largest species sample set I had was in